Okay, it is 12 o'clock. Time for our Facebook Live event with Dr. Richard Boles. My name is Kimberly Ruckman, and I am here. I work as a social media consultant for NeuroNeeds, and I'll let everybody else introduce themselves.
Dr. Boles?
Hi, I'm Dr. Richard Boles. I'm a pediatrician and a geneticist. I was 10 years at Children's Hospital, in 20 years in USC, and I have worked in industry, and now I'm in private practice, and I am one of the people involved in NeuroNeeds, one of the partners that makes this is being given to you by NeuroNeeds today as a company that makes products for people with neurodevelopmental disorders and other neurological disorders.
Hi, I'm Nick Gora. I'm also a principal at NeuroNeeds. It's a pleasure to meet you all and welcome to our Q&A. I just wanted to let you know that we're really interested in making sure that you understand how our products work, the science behind our products, so you have a better understanding of not only our company and our approach, but also the very products that you may be using or are interested in. And so the first part of this Facebook Live, I'm going to be walking through an infographic with Dr. Boles to help everybody sort of better understand what are pretty complex topics. And along the way, we're going to open up the discussion to some questions. So you'll see on the infographic in sections 2, 4, and 5, we're going to open up. We're going to take a little pause and see if anybody has any questions, needs some clarification. And then at the end, Kim's going to take over and just handle your questions and deliver them to Dr. Boles. So 3 takeaways that we want to make sure everyone gets from this discussion are that autism is genetic, autism is treatable, and the two are not mutually exclusive. And what that essentially means, there's hope that you're not stuck, that with the use of modern science, we can help identify some of the causes of autism and treat them. So we'll put up the infographic. You'll all be able to see this. And I note that we'll put a recorded version of this on our YouTube page, so you can go watch this again if you like. And that we have a blog article called Autism is Genetic, Autism is Treatable. The two are not mutually exclusive on our webpage. That is a summary of an article Dr. Boles published. And the link to that published article is also on our blog page. So we'll put up the infographic now, and we'll walk through these steps, and then we'll open this up to your questions. Do you want to put up the infographic, Dr. Boles?
Okay.
Okay. So again, we're going to stop at the end of number 2, number 4, and number 5. And if anybody has any particular questions or needs some clarifications, we'll do our best to answer. So as you can see from the first item, factors leading to disease. Dr. Boles, do you want to tell us what those 2 factors are? People can see them. It says every disease has a genetic and an environmental component.
Yeah, well, I mean, that's true, is that genetic and environmental components, that's pretty much everything that will cause disease. But every disease has genetic and environmental component. Having said that, it's not 50-50 in every disease. Some diseases may be 10% genetic, others may be 90% genetic. Most of them are somewhere in between. People will say, well, what about Down syndrome? Isn't that 100% genetic? No, 2 children with Down syndrome can have the same 21st chromosome, but environmental components can make a big difference, such as the early intervention in childhood, education, motivation, family life, etc. There's a lot of nutrition. A lot of things can make a difference. And maybe that's, 80, 90% genetic. And people will say, what about an automobile accident? I mean, isn't that entirely environmental? That can't be genetic. Go, no, that's genetic as well. People that have certain genetic traits that make them more daredevils, that are less afraid of risk, are more likely to drive fast, more likely to drive a lot, more likely to drink, more likely to drive and drink, et cetera, et cetera. and they're overrepresented in crashes and injuries. So the fact that everything is genetic and environmental, when people say, well, what's the cause of autism? It's genetic and environmental. Neurodevelopmental disorders, of which autism is one example. There is also ADHD, intellectual disability, epilepsy, et cetera, et cetera. Those neurodevelopmental disorders are genetic and environmental as well. They tend to have high genetic components, but they also have important genetic components. So the point of number one is that the neurodevelopmental disorders are like every other disease.
Okay, and let's talk about one particular topic that we'll get to later, homeostasis, because I think that's an important concept to understand relative to disease. So can you tell us the relevance of homeostasis to these particular factors?
Well, homeostasis is balance, and that's in many ways an Eastern philosophy that disease occurs when the body is out of balance. Think about the body as many different pathways. One would be energy metabolism, another would be neurotransmission, et cetera, et cetera. When these pathways are in balance, there are many different aspects are kept within certain degrees. Like the sodium concentration can't be too high or too low, the calcium, et cetera. Then you have homeostasis. Now, nobody's genetics is perfect, and some people's genetics put strain on homeostasis. And certainly nobody's environment is perfect. And some environments put strain on homeostasis. And when there's enough of a strain on homeostasis, the body may not be, the cells may not be able to compensate as well. And they try to compensate as long as they can, but at some point there's the straw that broke this camel's back. They can no longer compensate. They fall outside of the recommended range or the healthy range. And then it converts. And then you have disease, or you have, I should say, risk for disease. Because let's say a bunch of teenagers are staying up late and drinking and otherwise not really doing good health, eating pizza and other things. So those things are going against the homeostasis, but if one of them had a viral infection, even a mild one that they didn't know about, they could all get sick and be in bed for a week. But if one of them didn't have a viral infection, they may have gotten away with it. You see, the body tries to avoid perpetrations to homeostasis, tries to bring it back into balance. But if the genetics plus the environments are too much, then you're outside of that. You're at risk for disease.
Okay. So let's, in item 2, talk about one of the factors, the genetics. So you talk about, you make 2 points in terms of the genetic components of disease. There's the inherited genetic background, Can you explain what you mean by that?
Well, the difference between the two of them is genetic is not necessarily inherited. Genetic means it's written in the code of the DNA. It can be inherited, that code could be inherited from one or both parents, or it could be a new mutation that's not inherited. Almost all of the time we think of genetics as inherited. And for most disorders, not autism, for most disorders, we're talking about inherited changes. Again, usually from both parents. Multiple changes, not just one mutation, say, one variant in one gene, but many, many variants, many of which might be somewhat common and benign in themselves. But combinations of those variants and multiple genes can confer risk or can confer the opposite. They can be vulnerability factors, or they could be factors that are protective against disease. So the genetic factors will show you how likely it is to go outside of homeostasis and how likely you are to get disease. So, I mean, any disorder that you can think of is likely polygenic with multiple genetic components, usually inherited. Diabetes for one, asthma for another, hypertension, high blood pressure, any of these. So let's use an example of diabetes because almost everyone has somebody in their family that has it. People know about that. It's mostly genetic. It runs in families, but there's a very strong genetic component. There's a very strong environmental component as well. If you have the genetics for diabetes, yet you keep your blood pressure good, you eat well, you exercise, you can mitigate it or maybe even get rid of it without medication. On the other hand, if you have a family history that has very little diabetes, but you eat a very poor diet and are way overweight, you could be diabetic. So it's a combination of genes and environment. And that's what really affects all disease. And in autism and other neurodevelopmental disorders, you do have genetic background. That is important. In many of these families, you'll get other neurodevelopmental disorders like learning disabilities or ADHD in a few family members, and then that shows you that there are genetic components that are conferring risk in that family.
All right, but you also mentioned de novo variants, which are common in NDDs.
Yeah.
So what did you find in your study in terms of these de novo variants and autism in particular?
Well, let me explain what the de novo variants are first. De novo just means new in Latin. These are new variants which are not in the parents. What we did is we did whole genome sequencing, that's sequencing all the chromosomes and the mitochondrial DNA from end to end, sequencing the entire 3 billion base pairs of all of our DNA in our cells. That's much more than, let's say, whole exome sequencing, which is sequencing all the genes that's done in most academic centers. That's actually only about 1% of the DNA. We sequenced over 99% of it, there being tiny little areas that are still hard to read. So when you look at 99% plus of the DNA, you can do that in both parents and in the child. The child could be an adult, the patient. And we used a computer to subtract the mother's and the father's genetic sequence from the child to determine what was truly de novo. And then we looked at that. So the study was 50 patients, which makes it easy to put it into percents. And in 50 patients, we found 34 that had a genetic variant that met a very large number of criteria that I'm not going to go into right now, such that it is extremely highly likely that it is the major genetic component of the disease. In other words, we found the genetic cause of disease in these patients. That's 34 patients, 68%, more than 2/3 of them. So 68%, we found a genetic cause of disease. 50 of those 68 were de novo. They were not in the parents. It was a new mutation. And then 18 of them were inherited.
So de novo variants can play a significant role in identifying the risk for autism.
Yes, I think that it's higher than 50%. There's a lot of them that we had to eliminate because they didn't meet our very strict criteria. For example, that we used conservation, how likely it is, how often it is in other species. We used that it had to be associated with an autism gene, and then there may be new causes of autism that we were discounting. We're doing now 100 patients of Dr. Richard Fry. I've looked at the sequence at 85 of them so far. It's looking more like 60% or so. But the things here is that the vast majority of patients with autism have major genetic components. So not just genetic background offering a little risk. And most of that risk is de novo, not in the parents, which explains why you have kids with autism in unaffected parents and unaffected families in many cases.
Okay. Now, Kim, do we have any questions up to this point?
Well, I do have one question. What causes de novo variants then?
Very good question. We're going to get to that in number 4.
Okay. And that's it for right now.
Okay.
Okay,
All right,
one thing
good.
I want to mention.
But I do want to mention to everybody who's watching right now, if you do have a question, leave it in the comments below and we will get to it either during the presentation or towards the end.
Okay.
One thing I want to mention, the de novo variants, you don't find them like in diabetes and asthma and hypertension. You find them in neurodevelopmental disorders and syndromes. What I mean by syndromes would be like birth defects, and often people with birth defects do have neurodevelopmental disease as well. So those are the things in which you would need to do a trio sequencing the parents as well. For the vast majority of disorders, it's genetic background that's inherited, but autism has this unusual component. Only that and syndromes are really often de novo.
Okay. Now let's go on to the next one. The second part of our factors of disease is the environmental components of disease. We talk about diets, toxins, and many other factors. Can you elaborate on that a little bit?
Genetics and environment is in everything. It's not a zero-sum game. Every disease is genetic and every disease is environmental. People think it's one or the other. If you have more of one, you have less than the other. A better way to look at that is it's all of both. Now, the paper that we're talking about, I'm a geneticist and the paper was on the genetics. That doesn't mean that environment is really important. There are other doctors that work on the environment and they write about the environment. Some of the more important environmental factors are in the diet. Multiple aspects of the diet are important in terms of the components or factors leading to autism. But there's also toxins. There's toxins in the diet, in the water supply, in the air, the plastics that we use in daily life, the cadmium and other heavy metals, which are in the soil, things that we get medically, such as Tylenol and many other things. These things are components which can contribute to disease as well. So when I'm talking mostly about genetics as a geneticist, I'm totally aware of the fact that environment is really important.
Okay, and that leads us into item 4, which is the interaction of the environment with the genetics. So for example, you note environmental components regulate genetic expression. That's kind of a complicated concept. And in previous writings, you've given a sort of bridge or book on the shelf example, and that may be helpful, I think, to explain what you mean by this.
Well, let's use the library example. Let's say that there's a university library and there's a bunch of dusty old books that are sitting, and there are indeed hundreds of thousands of books that are sitting in the library. The vast majority of them are just on shelves that nobody's touched for a while. And then you have students going through from time to time as they need the information in those books, pulling a book out, sending it to the photocopier, and then leaving the library with the photocopy while they put the book back on the shelf. That's really what genetics does. The DNA in our nucleus is the library. It doesn't do anything if it's not read. It just sits there. It's maintaining the knowledge base for the cell. If the cell wants that information, it needs to copy it into RNA. And then it needs to take that RNA out of the library, out of the nucleus, where it then goes to do something. Apparently, these students are photocopying it not because they're just going to put it in a notebook, but they're going to use it for some purpose. Let's say that it's their chefs, and they want to use it to make some sort of culinary masterpiece of some kind. So they go get the right cookbooks, they copy the recipes they want, and then they take them out of the library into the kitchen, where they can then use that information to actually make something in the real world. In genetics, what it does is that the RNA leaves the nucleus and makes a protein. Proteins are the workhorse of the cell. They're the major structural components, the enzymes that do the chemical reactions, the ion channels we'll talk about later that regulate salt balance. All of these are proteins, and the body makes the proteins as it needs it. So in this analogy, unless you read the gene, it just sits there. So it's really reading the gene that's important. And what makes people go in and out of that library is their need. But people are out there, the environment, the people in the restaurant are ordering food. And then so they have to run around and get these things made. It's the environment which gives the signal for the cell to make proteins. So even as a geneticist, the environment's extremely important because it's the environment that's going to be used to turn all the genes on or off. Most of the genes being off in any given cell at any given time, but the environment will turn genes on. So okay. I cannot change what's written on the shelf, but what I can do is send students in to copy them or not send them in.
Mm-hmm. Okay. And the next item, you note that environmental factors affect genetics by increasing de novo variants. Now, you've mentioned that de novo variants have played a significant role in NDDs, neurodevelopmental disorders. So that suggests that environmental factors can increase the risk of NDDs as well.
Well, it appears that the de novo factors are extremely important in who gets an NDD, like autism, and who doesn't. If you notice, it says on here, Dr. Boles proposes. I cannot think of another reason that really, another explanation that fits the data, and it fits the data completely, is that in patients without autism, patients without any neurodevelopmental or syndrome disease, they almost never have a de novo variant that changes a coding sequence, maybe about 0.2. So 10 patients, you'll find 2 of them that have a de novo variant that change a coding sequence. This is less than 1. On the other hand, inherited variants are about 50,000 per person. So each person has about 50,000 inherited variants and far less than 1 de novo non-inherited variant. So those are special, those are unique. I don't see them very often out of autism. There's been many papers that have shown that. But in autism, I find in most of my patients, 1, and that usually is the cause of disease from my investigation. Occasionally, I'll find 2, 3, or 4 even. Some patients have more than 1 de novo variant that I think is contributing to the disease as well. So why are all these de novo variants there? Maybe they were always there and we just didn't know it. But I think that, and that's possible, but I think it's also likely that our mutated environment, our chemical-laden environment, is causing these mutations, or at least increasing them dramatically. And there's so many different things it could be. There's a lot of chemicals in our environment that we know mutate DNA, and that's not what this talk is about, but it is fairly probable. That would explain why genetics is that autism is predominantly genetic. That's the first thing I want people to understand. Why? Because it's true, and to really understand how to treat them, you need to realize that. And number 2, that autism is increasing dramatically because these mutations are increasing dramatically as we increasingly alter our environment with toxins.
Okay. Kim, do we have any questions coming in on
should I use the bridge analogy first?
Yeah, I think it would help people for you to use the bridge analogy in the context of especially the gene regulation, gene expression.
Okay. Well, the idea is, remember that we really can't change what's in the DNA. That was there at conception, and it was actually there in the sperm and the egg before conception. And yeah, there's genetic engineering, but that can do a teeny bit now, and that's not something that's practical, particularly not for something in the brain at this point. So we can't change what's written on the book, but we can send students in there to copy what we want or not send them in to copy. And that is manipulating the environment to alter genetic disease. So I used an example of a bridge. Let's say a de novo mutation, like a ship, hits a bridge and the bridge goes down, okay? That bridge is down. We're not fixing that bridge. It's down. But now you have a problem with homeostasis. You have cars backing up all over the city. So not only the people that want to cross the river, but people just want to get home from their job on the same side of the river. They're in gridlock. You have disease now. You have a disruption to the homeostasis of the city, of the traffic flow. Well, one thing that we can do is we can open up other additional bridges, additional lanes on other bridges to get the traffic out. And that's an example of a genetic defect. What can you do about it? Well, if you understand what the genetic defect is, you can manipulate the environment, not to cure it, but to treat it, to make it better, to improve the patient.
So help me understand this a little better. So essentially the cells aren't able to do what they need to do. They're not able to perform the functions they need to do. So you manipulate the environment, you create that new bridge so they can carry on with the functions the body is demanding at that time.
Well, we figure out what the genetic defect is, usually by sequencing. And then we say, well, how would that cause problems in the cell and in the body? How is it causing the symptoms, the disease, the problem parents are talking about? And how can I intervene in a positive way?
Okay, Kim, do we have any questions on this item 4? Any issues people are having with the explanation?
Yeah. So you mentioned turning on and off genes. How would you turn off a gene?
Well, the genes are generally off on their own, and they turn on when they need to be turned on. Sometimes we can turn off a gene by just giving the product. That say A plus B is converted by an enzyme to C plus D. And that gene is, let's say, is not working correctly. It's on too fast or whatever. One thing we can do is we can just give a huge amount of C, and the body will say, oh, we don't need to make any more C, so we can turn the enzyme off. I mean, there are things like that. That's a biochemical intervention. I do that a lot. So if the problem is that the gene is on too fast, not every gene is destroyed like a bridge. It could be a gene that's destroyed. It's certain that it's on too fast. We can give the product. Another thing, if the gene is going too slowly, we can give like A. We can give a whole bunch of A, and then it's just going to go to get more C. Or we can give more B, and it's going to go to get more C and D. So those are like examples in which we can manipulate the system to turn on and off genes. We don't always have to turn on and off genes. Sometimes we can just, if say that the enzyme doesn't work and you can't make C and we really need C, we can offer C in the diet or as a supplement.
So supplements, medications, things like that.
I mean, what do we actually do? It's mostly supplements, and often at extremely high dosing because we want to really manipulate it to really move it where we want to go. But sometimes it's other things. It's really medical care. I mean, what can you do as a physician? You can offer drugs. Sometimes they are prescription medication. You can offer diet. You can offer exercise. You can offer supplements. I mean, sometimes it's other things, but those are generally what we're talking about.
Okay. Somebody asked, and I guess it goes more to the environmental components, about sickness/illness triggering autism or neurological disorders. Like the kid was fine and then they became sick with something and then they started exhibiting autistic behaviors.
That's actually a very good question. It's the homeostasis, and the cells will try to maintain homeostasis as long as possible. No one has perfect genetics. No one has perfect environment. Everyone has factors which are trying to push it out of homeostasis. Life is very redundant, has many different pathways, and it's very resilient. But at some point, you just have, like I said, the straw that breaks the camel's back. The metaphor is the camel is doing fine. You keep adding more and more. At one point, you put one more straw on, and the camel falls down. That's really often what it is with homeostasis. The cells can handle it until a point they can't, and then they fall apart. They actually can just do the wrong thing, or they can even die. A viral illness can be that last straw. Even a common viral illness could be in a cell that's just barely getting by, could no longer get by. There are many other things that can do that. I've seen medications do that, such as steroids and anesthesia. Unfortunately, it's true. I've seen vaccines do that before. I've seen it with people that are up all night and eating poorly. I've seen it with alcohol before. There's a lot of things that can hurt homeostasis. Tylenol, cigarette smoking, or even secondhand smoke, not sleeping right, exercising way too much, like doing a marathon when you're not trained for it. Or you've done high school sports and then now you're doing nothing and you're a couch potato. But all of these things are perpetrations on homeostasis. It's really, what your mother and your grandmother have always been telling you, good, clean life. Those are things that help maintain homeostasis. No one has perfect genes, but you can try to reduce the factors as much as possible.
Would since you bring up relatives, grandparents, things like that, would how they ran their life, say you had a grandparent who was an alcoholic, would that affect the genes for the upcoming generations?
There are epigenetic changes that might cause issues, and those are certainly important in things like diabetes and obesity. Hypertension, we're not really aware of those things as much in the neurodevelopmental disorders. There are mutations in the genes that are involved in epigenetics, the mutations in the genes that actually regulate epigenetics. Those can cause autism. I've seen those several times. But what kind of factors occurred in previous generations? Well, certainly chemical exposures to boys that affect the mutations. that will then be in the sperm and eventually be in their children. Those are definite. I think that those are the main cause of the de novo mutations. It's just a true thing that autosomal dominant new mutations are almost always from the male, and chromosomal abnormalities are almost always from the female. It kind of balances out, but these new dominant mutations, about 90% of the time, do come from the man.
All right. I want to stay on track if there are no other questions on this point. And Dr. Boles, because the next point about disease results from disturbances of key pathways, first, I'm presuming you're talking about key pathways relative to neurodevelopmental disorders.
Yes.
Okay. And these disturbances, again, we go back to homeostasis. They're, I presume, on a level that is impacting the homeostasis of functionality here.
Well, homeostasis is a balance in all of the pathways, and there are a very large number of pathways, but the pathways are still the bottleneck. There are many hundreds of genes that have been shown to cause autism. I suspect there's far more than 1,000, probably more like 2,000 or 3,000. There are thousands of different ways in which a kid with autism can present. But in the middle, there's really only maybe 10 or 15 pathways. In my mind, in my experience, half those pathways are treatable. Now remember, every pathway has to be in homeostasis. So you need a balance in each and every part of your life, not just a balance in one part and off on the other. I've mentioned 3 pathways down here. Why? Because these pathways are common in neurodevelopmental disorders. And so a lot of people have problems in them. And number 2, these pathways are highly treatable in my experience. I treat them all the time in my patients. This week alone, I've treated all 3 of these in my patients. So they're mitochondrial function, cations, and neurotransmission. And before I go into each one, let me just explain how they kind of fit together in a model.
Okay.
So the way that the nervous system works is you have a nerve cell, a neuron, and it's not connected to the next neuron. There's that gap in between, right? There's that synapse. So the signal has to go from one side of the neuron to the other, and that could be several feet long, like to get to the toes or to snake for the intestines. And then there's the synapse that has to cover that gap. So ion channels are involved in sending the signal from one end of the neuron to the other. Neurotransmission is involved in sending the signal from one nerve cell to the next nerve cell across the synapse, and it's all powered by the mitochondria. So that's how these 3 pathways are separate pathways, but they're all interrelated to each other, and why the brain is really a complex electrical circuit of neurons and synapses, all powered by mitochondria.
Okay. Why don't you go over each one briefly? And again, in the past, you've used metaphors to help explain their function. And then we'll tie it into treatment.
Right. Okay. So mitochondrial function, that's the battery, the electrical outlet in your house that powers everything in the cell. The mitochondria make over 95% of the energy the cell needs. Energy is life. The difference between somebody who's just alive and somebody right after they're dead is one's energized and one's not. Without energy, there's death. Energy makes, every cell needs energy to do almost everything that it needs. Cells need a lot of energy, but nerve cells are electrical and they need more energy per unit, like per ounce, than any other tissue, other than muscle that's heavily exercising. Yeah, that needs a lot, but other than heavily exercising muscle, nerve uses the most. So that's why mitochondrial dysfunction predominantly causes neurological disease. Think about a car. You have an engine of the car. That's like its mitochondria. It's burning fuel. What's the fuel? It's gasoline. Gasoline is almost identical to fats that are in our diet. But anyway, you burn the gasoline, you make the engine for the car. The thing is, the car has one mitochondria, one engine, where the cells have thousands for each 1 billion of the cells. So it breaks down there. But let's say that the cell is, the car is going up an incline, up a mountain, pulling a trailer, and it's going fast. And let's say that the car's engine's not really been finely tuned. And it's not going to do very well. It's not going to have the energy, and it's going to produce a lot of pollution because you're making the engine burn too much fuel. it's going to burn some of that gasoline only halfway. Half-burn gasoline is chemically the black smoke that comes out. It also coats the engine and damages the engine even further. So you'll have even less power after you do that for a while. So the same thing in the body. If the brain is needs a lot of energy, and let's say it's under stress, such that a viral illness or any psychological stress, and in somebody whose mitochondrial DNA is not perfect and they're not making a huge amount of energy, they're on the lower side of the normal range, you can see that mitochondria is going to be stretched. It's going to burn halfway through fats and sugars, and those are going to produce the lactate and the organic acids and the reactive oxygen species and everything that we can find in mitochondrial disease. Those are damaging to the cell. They damage DNA, they damage proteins, they damage membranes, and they damage the mitochondria itself, so it even makes less power. So then eventually you get out of homeostasis and you get disease. So because the brain needs more energy, the mitochondrial dysfunction predominantly affects, well, not just the brain, but the whole nervous system. And when does it predominantly cause problems? During stress. What is stress? Stress could be psychological stress, losing a relationship, failing a test. It can be physiological stress, hot weather, fasting, viral illness, alcohol, etc.
Okay, the cation channel. This is a complicated one, and I like the example you give of a dam, how it works.
A lot of people understand that mitochondrial function is important in autism, but the cation channels, unfortunately, it's not nearly as well understood. The cations are, what a cation is a positively charged salt. And I'm going to talk mostly about sodium and calcium, but also potassium, magnesium. There are many others. Okay, so sodium and calcium are positively charged salts. The channel is the gateway in the membrane to let it through. These salts cannot go through the membrane because they're polar, they're charged, unless they're invited in. Basically, a channel opens up. So you have a calcium channel, and it lets in calcium when the cell is triggered to let the calcium in. It's very, very complicated when it lets it in. But what if that calcium channel is leaking? Because there's a mutation in the gene, the protein's not perfect, and it doesn't quite close well enough, or it stays open a little bit all the time. Then you have calcium, which is constantly leaking into the cell. Calcium is positively charged as a cation. When cells are positively charged, they're energized, they do their thing. Nerves send impulses, muscles contract, glands secrete hormones, etc. When any cell is positively charged, the cell is, okay, I'm on now, I have to work, I have to do what I'm supposed to do, otherwise it sits there. So the leaky channel causes the cell to be on, causes the cell to be hyperactive. That's going to cause problems in and of itself to homeostasis, and that can result in disease. But you also need to pump all of that calcium back out, and a hyperactive cell needs more energy. So here goes the dam analogy. You have a dam and it's holding back a lake from a river. If you have a hole in the dam, the water's going to come through. Eventually, it's all going to go through, but then you have pumps to pump it all back. Yeah, if you have enough pumps, you can keep the water on one side of the dam, but that's going to use a huge amount of energy. And that's why cation leaks cause mitochondrial dysfunction. Not because you don't make enough energy, because you use it too quickly. But also remember, you have an energized cell that's positively charged because of the leak that needs more energy. So defects in calcium and cation channels, particularly calcium channels and sodium channels, I see those very frequently in autism. And that tells me where to treat it. I like to see those because those are treatable.
Okay, now neurotransmission. Why don't you tell us what neurotransmitters do, and then we have excitatory and inhibitory transmitters, and you've used various metaphors for those as well. I think these metaphors are really a good way for people to help understand these complex scientific issues in context. So,
well, everyone probably knows from high school biology that you have one nerve cell and the impulse goes across that nerve cell. By opening ion channels, that's how it does it. And then it gets to the synapse, and to cross the synapse to make the next nerve cell fire, it has to release a neurotransmitter, which then binds to a receptor on the next cell. Okay, that's true, but most nerve cells in the brain have 10,000 other nerve cells that synapse onto it. So a synapse will have 10,000 neurons onto a single nerve cell. That's the reality of the brain. Some of those will be excitatory, that will, when they release excitatory neurotransmitters like glutamate, and some of them will be inhibitory. They release inhibitory neurotransmitters like GABA. But the thing is most of them are inhibitory. And I use the toddler in the cookie jar. If you have any small children, you'll know this one. So toddler says, I want a cookie. Mom says, no, I want a cookie. 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 Okay. Or the mother just walks out of the room. That cookie is gone. You have to constantly be saying no to take sure that cookie is not taken. So the excitatory neuron is going to take over and take that cookie if the inhibitory neuron, the mom, is not constantly saying no. So you have a synapse and the inhibitory neurons are firing no, 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 no all the time. When those inhibitory neurons go down to a certain degree, the excitatory neuron will take over and the next nerve will fire. That's how it works. But think about it. What's going to run out of energy first? The inhibitory neurons that have to say no, 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 no all the time. So when you have energy deficiency, you have hyperexcitability. And that's why I've heard this at least 100 times, mostly by mothers. Well, if you're saying my child has no energy, why does he have so much energy? Because it's the inhibitory neurons that run out of power first. So you can see, I said that the mitochondria power the whole thing. When the ion channels open to let the calcium or the sodium in, the mitochondria pump it all back out. And the mitochondria need to power the synapse as well to get all of those, particularly to power those inhibitory neurons to constantly be saying no. So they all are interrelated to each other. If you have a neurotransmitter problem that you have too much excitation, that's going to put problem on the mitochondria because now you have a cell that's hyperexcitable that needs more energy and perhaps it can't make that much energy. So you can see everything's synergistic. If you were an athlete and can make a huge amount of energy, ATP, you might be able to get away with that. But if you're more even in the normal range, but more on the couch potato side, you just can't make that energy. So normal variants in the population do make a difference here. And these three concepts, mitochondrial function, cation channels, and neurotransmitters, all interact in the very complex thing that we think of as the nervous system or the brain or life.
Okay, great. And you note they're very treatable. And I think you can sort of hear the underlying thread of energy deficiency through all of these. And so when you say these pathways are very treatable, now let's move into number 6.
We're going to open it up to questions first, are we not?
Yeah, excuse me, you're right. I got too excited there. I wanted to make a point, but Kim, are there any questions coming through on this number 5?
No, but I did want to mention, because the questions that we did have, we can't talk about individual circumstances so much, like individual genes and their expressions. I would say, if a person did have questions about those, to make an appointment with Dr. Boles or a doctor near them.
Mm-hmm. Yeah, that would be the way to go.
Mm-hmm. Okay, so I'd say go on to number 6.
Okay, so we've got this underlying thread of homeostasis and the body not able to regulate itself and enormous demands on energies due to these imbalances, which leads us nicely into number 6 in terms of the pathways are treatable. And so what are the ways that we treat these pathways? And let's talk about them and say the mitochondrial function. How do you treat those pathways, and can you give, if they're nutrient-specific examples? And we'll go down the list of these 3.
I generally, in all of my patients, when I first see them, treat the main treatable pathways. And these 3 that I've mentioned before I definitely think about. But there's also other treatable pathways that we didn't talk about here, because it would be a 5-hour-long talk if I talked about all the pathways. So methylation, yes, treatable, yes, important, not in the top 3. Inflammation, maybe that should be number 4, but we tried to simplify it a little bit. Neural inflammation, very important. Any of the top 3 problems can cause cellular energy, can cause cellular injury, and that can bring in the immune system. And the immune system requires a huge amount of energy as well. Mitochondrial problems can affect that. Cations can affect that. So it's all interrelated. There are other pathways, amino acids, and a lot of other treatable pathways. So when we get into the products, it's to treat the 3 major pathways plus the other treatable pathways. And also there's some really minor pathways like creatine. Yeah, every once in a while I see a patient with a creatine biosynthesis disorder. So I put creatine in the solutions as well for those few people that really need it badly. But that's not one of the major pathways, because I might see it maybe 1% of the time. I want to make sure I handle it. But what I do is I start out with cocktails of with no alcohol in it. Cocktail means things mixed together. Alcohol is mitotoxic, if you haven't already heard that. And that's often with teenagers that will precipitate their first break, is when they experiment. But beside that, I see that many times. Is that I put the things together in a cocktail of many different things, because each and every one can do something, at least in some people, but it's not going to make a huge difference, each every one. You need the synergy between them. Mitochondria is an assembly line. And in the talks that I give, and I show slides, I show examples of assembly lines. You have that guy in the middle of the assembly line making cars. You give him his vitamins, he's not making more cars, he's making chaos. You need the whole line to go faster. You need to give all of the parts of the mitochondria the energy that they need, the cofactors they need to be able to make the energy and to detoxify themselves, like antioxidants, to remove those reactive oxygen species, because the mitochondria is pushed too hard. So I put in vitamins, minerals, cofactors, and antioxidants, and there's some chemicals that don't really meet any of those, like creatine doesn't really fit any of those, but they're all nutrients, they're all natural, they're all in food, in normal everyday food that we eat. But so what's the difference between that and food? Well, the dosing. Some of them are dosed 1,000 times higher than the RDA, which is like 100 times higher than if you ate the entire salad bar. That's to push the chemical reactions in the direction of healing, to push them really hard. The other thing is that they're activated. Many of the chemicals are methylated in the right places or otherwise activated, so that they will easier to work in anybody of any genotype. People talk about MTHFR and all those things. I want every genotype, no matter what they have, to be able to handle this and to do better. And also the bioavailability. A lot of the stuff that's bought over the counter is cheap stuff, and it really doesn't get into the bloodstream. It just goes out the other end. So it's good stuff in the right at the right amount and in the right concentration. So if you give too much of something and not enough of another, you can get an imbalance. So that's how I make these. Spectrum Needs is the first one we came out with about 7 years ago. It's 33 active ingredients. It's a mitochondrial cocktail. Most of the ingredients do that. A lot of the ingredients are also for cations, neurotransmission, creatine, nitric oxide synthase, arginine, methylation, inflammation, and many other pathways we really don't have time even to mention today. 33 active ingredients in a powder. It comes in lemon or berry flavored. You can mix it in any liquid to any amount if you want a stronger taste or a weaker taste. Then we have Energy Needs, which is very similar, but it's more for adults and adolescents, older children. It's a powder that's in a capsule form, 40 active ingredients. It's almost exactly the same. So I say choose one of those. I recommend for all my patients, they take Energy Needs if they can swallow, and if they can't swallow, Spectrum Needs. Energy Needs is a little cheaper per gram per day, and it's also more dose for adults, and it's a little bit better in that it has 7 more ingredients. But Spectrum Needs is fine too, and Spectrum Needs has a really high B vitamins. A lot of the little kids with autism do really well at, but some of the older ones have more trouble, so then I go to Energy Needs. That's a powder, but oils and powders don't mix. And I tried to put everything in one box, in one container, and it would work, but the bioavailability is a problem with the oil-based. And so the coenzyme Q10, which is extremely important because that actually is the chemical that electrons attach to go across the electron transport chain. Coenzyme Q10 is it's very large and has major bioavailability problems. Almost all of the CoQ that you'll get in an average drugstore has very low bioavailability. I always get blood levels in my patients. Quest Diagnostics will do it. But blood levels are low for almost all of the forms. So we private labeled Conica, ubiquinol QH form in limonene oil. That's in lemon peel oil instead of in soy oil. It's a very clean product that gives very high blood levels.
And Dr. Boles, it's an antioxidant also, so why don't you explain to people the benefit of an antioxidant, especially when the body's out of homeostasis?
Well, when the mitochondria are made to act much faster, which they will do in disease states, even if the mitochondria are normal themselves, they're going to produce a lot of free radicals, which is because they're producing energy too quickly. The free radicals build up faster than they can be dealt with. Free radicals are very highly damaging to cells. They can cause DNA damage. That might be some of the mutations. They can disrupt membranes and destroy cells. They can age proteins, etc. We need to give antioxidants. The body has antioxidants like glutathione, but at some point, homeostasis won't be able to be maintained anymore, and you need outside help. A lot of the stuff that people have taken all over the world for thousands of years, particularly Chinese and Indian medicine, are plants that are very high in antioxidants. The plants make it to survive infection themselves, by the way. And these high antioxidants are very helpful. So a lot of traditional medicine is taking antioxidants. But now that we understand this more, what we can do is we can remove the antioxidants from these plants and from other things as well. And then we can formulate them in a way that's highly bioavailable.
Okay. And then the fish oils as well are another component that...
You might know about omega-3s. Omega-3s are critically important. Cave people ate a lot of omega-3s. It's in the fungus, it's in the mildew, it's in the moss, it's in the dirt. They got a lot of it. They did not wash their vegetables. They just pulled it out of the ground and ate it. Fish will eat the plankton, or the krill will eat the plankton and get it. So people these days will get it from fish and krill, seafood. But if you're eating a lot of seafood, it's expensive, not everyone likes it, and also there's a lot of mercury in it, which is a problem. Factory farming does not give omega-3s. You need to get supplemented with that. A lot of doctors know about that, but unfortunately, a lot of them really don't. Fish oil is great. A lot of older people will take fish oil. A lot of people will take fish oil because it's good for your hair, skin, nails. It's good for cosmetic purposes. It's good for blood pressure, blood sugar, heart, all of that stuff. It's great, but it doesn't cross the blood-brain barrier. It's triglyceride bound. Krill oil will cross the blood-brain barrier. It's great for autism, ADHD, epilepsy, etc. But krill oil is much more expensive. To get the dose for cardiac, for heart health, you need to like $300 a month of krill oil. So I recommend taking krill oil and fish oil because they both do different things. There's also, by the way, krill oil is phosphatidylcholine bound. That brings it directly into the brain, but also phosphatidylcholine, or PC, and choline are very important for the brain as well. Phosphatidylserine is also important for brain, and that's in sunflower seed. I made Omega Needs, which is high-dose fish oil plus krill oil plus sunflower seed-derived phosphatidylserine to hit all of that in one product. It's good for mitochondria. It's good for many of the pathways. It's particularly important for inflammation. It's an excellent anti-inflammatory as well as it's good for brain growth.
Okay, so we go back to the autism is genetic. I hope we've made that point here through our discussion. and the most important part, autism is treatable, and these two are not mutually exclusive. And the key concept here is homeostasis, right? We're helping the body, we're helping pathways get back into balance when they can't self-regulate, when they're under attack.
We're trying to assist the body to do what it naturally does, but at some point it just can't do it anymore.
Right. So I think we're going to open now up to questions.
There's a couple more things that I think that we ought to put in here. First of all, we spoke about the treatment for the mitochondria. We didn't speak that much about the treatment for the cations. Mitochondrial things are important because of cations, you run out of power because you have to pump it all out. But also things like potassium, magnesium for different reasons are very important. So I will often give very high dose potassium, very high dose magnesium to my patients. And many times I'll use different drugs to turn off the channel that's leaking. Now, this requires an understanding of the individual genetics of the patient. And certainly, if I look at the genetics of the patient and I look at the raw data coming from the computer, not just what the report says, I'll know how to treat my patient because I know their entire DNA sequence. Not everyone can take advantage of that, but everyone can take advantage of the fact that thousands of people with autism have been sequenced. And we know what most of the problems are in most of them. That's not necessarily that it will be in your child, but it's a very high likelihood. And these products will hit all the major pathways and all the minor pathways that how I can think about treating them. So if you can get sequenced, get sequenced. If you can't get sequenced, we can still help. These products will hit the pathways that are probably important in you or your child.
Okay. All right. Kim, do we have any further questions?
Yeah, I do have a question. I'm going to generalize it. Okay, so people who are using the NeuroNeeds products that they've started their children on the products and now they're having issues with sleeping. What would be your suggestions?
I have never seen a significant side effect in any patient ever on any NeuroNeeds form, because there's all natural things. Occasionally someone will have a side effect that's fairly mild and it goes away when you stop it. It's reversible, but nothing's ever been irreversible. Nothing's ever been severe. The two most common side effects are nausea, which you can have with any vitamin or any supplement. So we recommend start low, go slow. So start with a much lower dose. You know your child, you know your own stomach. If prone to nausea, maybe start at 10 or 20% the normal dose. Work your way up over a few weeks. Nausea, not a problem. You can start at maybe a quarter or half the dose. Work over a few days. But that would be the first one. The other one's over-energized. Kids with more energy will have more energy. They'll get into more trouble like more normal kids. Some of that is actually good. They'll more exploring of the world. But sometimes they just feel like they're more over-energized. There's things that you can do about that. You can move it forward. Instead of giving it at bedtime, give it at dinner time or even at lunch or midday, the second dose. If you're having trouble with sleep, move that dose forward. Another thing that you can do is that you can reduce the amount. The amounts that are on the label is what I recommend in most of my patients. Some take more, some take less, but that's my usual thing that I recommend. Not everyone needs the full dose. Sometimes half a dose is even better in that patient. So it's every individual. So another point to put in here is talk to your doctor about everything that you do, not just NeuroNeeds products, but all supplements, everything that you do, your doctor should know about it. You shouldn't just be flying solo on anything. But the other thing you can do is you can balance it out. We have another product called ComNeeds. It works on the neurotransmitters. It helps GABA, the inhibitory neurotransmitter. GABA, L-theanine, which is from tea, and then B6 and magnesium all help the inhibitory neurotransmitter. But it also has 5-HTP, which helps serotonin, which is mostly an inhibitory neurotransmitter. When people take ComNeeds, it often will balance that out. So I'd say maybe 20% of people, when they take the full dose of energy needs or spectrum needs, that they're over-energized. You can lower the dose, go up slower, or balance it out with ComNeeds is what I recommend. Any of those things can be helpful.
Okay. Any
And
other
did
questions?
you want to explain what is in calm needs? That helps with that.
Yeah, I went for, there's the 5 ingredients, the 4 that help GABA and the 1 that helps serotonin. It will balance that, but the other thing it will do is most of my patients that take calm needs take it in the morning because of social, test, or school anxiety. For the vast majority of people, it does not sedate. You can drive a car, you can do a math test on it. I recommend, trying it before you decide to drive a tractor trailer, but, it's, be reasonable about it. But the vast majority of people, it just mellows you out a little bit, and you can go to school and you can learn that day. Some people need much less of a dose. We usually say 2 capsules in an adult. I usually give kids 1 capsule, but you start at a lower dose. Some people, 1/2 a capsule works great in them, and that's what they need, and more than that is a little bit too much. So start low, go slow, find the right dose for you.
I think that's it on this end. Again, we can't answer individual questions about, specific genes. There was a question about SNP data, 23andMe. What are your thoughts on that?
Okay. Well, 23andMe is a toy, and it's a fun toy. Like a little toy Lamborghini. It's kids like that. Even some adults like little toys, but it's not going to get you very far. The 23andMe is sequencing 1 millionth of the genome. I sequence over 99%, not 1 millionth. Some of that information can be helpful in the right settings, but I do recommend to go beyond that.
Yeah,
Okay.
so we're going to put up this talk on our YouTube page, and you'll also be able to see this summary with the highlights points, and hopefully that's something that will help you if you have any further questions or refresh your memory. And then I'd like to thank everyone for participating, Dr. Boles and Kim, for taking your time out, and all of you for taking time out of your day to listen to us. I hope this helped you better understand how our products work, the science behind them, and I wish you all well.